Okay.
Okay, well, it's my pleasure today to welcome Dr. Santosh Kumar, who is a professor and vice chair in the Department of Pharmaceutical Sciences at the University of Tennessee. Dr. Kumar is also an assistant dean in the College of Pharmacology. Dr. Kumar's laboratory investigates the neuropathogenesis of HIV and its associated comorbidities, including substance abuse disorders, neurological diseases, and co-infections. In addition to this work, his lab is developing innovative treatments for neuro-HIV using lipid-based extracellular vesicles and hybrid nanoparticles for targeted drug delivery. And I think he may talk about some of that today. Beyond his academic research, Dr. Kumar has also served as the president of the Society on Neuroimmune Pharmacology. So welcome, and we look forward to your talk.
Okay, thank you very much, Matt, for such a nice introduction. And hi, everyone. Good afternoon in the East Coast, I believe, already. And good morning for the rest of the country right now. So again, I appreciate Carlin for inviting me to give this talk on such a prestigious platform here. So as Matt said, today I'm going to talk mostly on my most recent project on novel drug delivery modality to suppress HIV neuropathogenesis, including hand. So as I said, we have been working for last over 15 years on different HIV-associated comorbidities with alcohol and with tobacco smoking. And we have connected many of the molecular basis for these neuropathogenesis in drinkers and tobacco via cytochrome P450, oxidative stress, and also extracellular vesicles and exosomes. But more recently, we started working on treatment strategies for HIV neuropathogenesis and hand. So that's what I'm going to talk today. So as most of you know, it's not unheard of this audience that HIV infects brain cells early, very first or second week of the infection. It can package inside monocytes or macrophages, and using Trojan horse mechanism, it can enter into the brain and then reinfect perivascular macrophages, microglia, and to some extent astrocytes. And once these macrophages, macroglia are infected, they produce neurotoxic agents, including viral proteins, and that can activate macroglial cells and then produce inflammatory response. And ultimately, these can lead to synaptodendritic pruning as well as neuronal injury and ultimately HIV-associated neurocognitive disorder. So if you look at the data on neurocognitive disorder, even though during the COT era, the HIV-associated dementia has reduced, but overall, hand prevalence is still same or even higher than the pre-COT area. And as we know that HIV can also accelerate aging by almost 10 years, and we have over 50% of the people who are 50 years and older. It means they are now getting at the verge of getting other neurological diseases, for example, Alzheimer's disease, ADRD, stroke, and others. So clearly, it's not about just hand, it's about HIV-associated other neuronal diseases, which are going to pose a big risk in the future. So to begin with, we have to find a way to treat hand. Because once we do that to some extent, we can reduce even other HIV-associated neurological diseases. The challenge with treating hand is two major challenges. One is subtherapeutic concentration. Most ART drugs do not enter into the brain to the therapeutic level. And also, even though they do, some of them do, they can cause neurotoxicity and neuronal damage, and that may to some extent also cause hand-related pathologies. So how to overcome these challenges? So these are the three different strategies our lab have been using for some time now. And the first is to find a routes for administration which can overcome these two challenges, which are, brain drug concentration enhancement and also for neurotoxicity. The second is use of some kind of nanoparticles, either synthetic or exosome-based or maybe even hybrid. And the third is perhaps nutraceutical agents and adjuvant, because once we use adjuvant, we may reduce the drug dose, which may reduce drug-induced neurotoxicity. So these are three strategies we are thinking to use for our purpose. So to begin with, like the selection of different ART drugs. Of course, there are many drugs one can use, from most recent drugs to even earlier drugs for repurposing. We have been studying with these drugs quite a bit in the last few years. Our two postdocs have started working, and we saw the interaction with alcohol, tobacco, etc. So we are trying to use these drugs, and also because they are well-established ART drugs, either in the past or some even current. They have good to average CNS penetration effectiveness score, called CP score, and also they are relatively less neurotoxic. So those are the three kind of general reason why we wanted to choose these drugs for our study. So in terms of very first study, we established that intranasal drug delivery is more effective than intravenous. And just showing you one data, that when we did this study in wild-type mice at both high dose and low dose, you could see that the intranasal delivery can enhance drug concentration in the brain, mainly early in the time period at both high dose and low dose. And it can reduce the concentration to some extent in the other organs like liver, lungs, and plasma. So all these can say that, yes, intranasal drug delivery may be beneficial for increasing brain drug concentration and reducing off-target effect. So second thing is the nutraceuticals. We know that many of these nutraceuticals are being used either in clinical trial or for research purposes for many diseases, cancer, and some neurological diseases as well, including Alzheimer's, Parkinson's. So we thought we can try some of these which have inherent characteristics as an antioxidant and also anti-inflammatory. And these are the hallmark of HIV-associated neuropathogenesis. We have also studied some of these nutraceuticals and the analogs, like resveratrol analogs and cucurbitacin D, and we have shown that these can also have anti-HIV activity. So they not just have antioxidant and anti-inflammatory, which could be useful, but also they have anti-HIV activity. So we wanted to use some of these adjuvant nutraceuticals along with our ART drugs. So very first experiment we planned, we looked at whether curcumin can enhance elbitagrevir concentration in both in vitro and in animal system. So in the case of in vitro system, using HIV-infected human macrophages, it's a cell line, which are a cell model for HIV study. And we could see that there's an increase in EVG concentration in the presence of curcumin at early time point or medium time point, 10 to 24 hours. And overall area under the curve is also significantly higher. When we look at some of the pro-inflammatory cytokines, and we could see that there's a reduction on these cytokines, especially when we use a curcumin and elbitagrevir together, like the fourth bar you see. There's a significant decrease in some of these inflammatory cytokines, which are important and are induced by HIV. We could also verify that at the protein level, mainly for IL-1 beta and TNF alpha. Then we looked at the EVG concentration in the brain in wild-type mice, and what we could see that the EVG concentration is again significantly increased in the presence of curcumin in the plasma You look at the area under the curve, clearly increased with IP and also with IN. And if you look at the brain concentration in both the cases, but IN clearly had a bigger increase compared to IP for EVG concentration in the brain. It also increased in lungs and to some extent in the liver as well. But clearly there's an increase in EVG concentration in the presence of curcumin, not just at high dose, but also at low dose, which I'm not showing you the data right now. But we have published this in scientific reports last year, a year before. So we also looked at some of the neuronal markers just to see that whether neuronal homeostasis is preserved. And what we could see that at least these 4 markers we looked at for neurons, for macroglia, and for astrocyte, all these are unaffected. So they are not altered in the presence of either EVG, curcumin, or together. So then we moved on to doing some of the pharmacodynamic study using HIV mice, and we chose to use eco-HIV mice for a number of reasons. First of all, this mice was produced or generated over 20 years ago by Walsky group, and they have done quite a bit of work, even recently as well. So in this mice, the main change is that the human GP120 was replaced with animal or mice GP80, so that it can be infected by HIV. And what they have shown over the years that these infected eco-HIV can have infected macroglia. They can also activate and enlarge macroglial cells. They can damage neurons. It can be seen by reduction in synaptophysin and impairment in dendritic density and axonal structure. As well as these mice have hand-like effects. So clearly, again, there are other humanized mice model as well for HIV, and that can also be used. But at least for our purpose, we felt that this is the most appropriate animal model because of these reasons we just discussed. And more recently, there are a lot more, many papers are being published on this model. Now, so in terms of behavior study, we followed 3 different studies, novel object recognition and Morris water maze, which are already well-known behavior study for hand. We also included catwalk gait analysis, which is more like a motor function or function for balance. So we included the third one, catwalk, which is usually studied for stroke and Alzheimer related behavior characteristics. So if you look at the first data on node analysis, again you can see that in HIV mice, the recognition index and discrimination index both are reduced. But in the case of EVG and curcumin and mainly combination treatment, they are rescued. They pretty much come back to the wild type or uninfected mice level. So that was pretty good finding we felt we got from this combination. Then we looked at more water maze analysis. We looked at escape latency and distance traveled. And what we can find mainly in the early time point, eco-HIV had increased escape latency compared to wild type mice in the bottom. And the treatment, mainly combination treatment, have reduced to almost to the level of wild type. The same thing happened with distance travel as well, that in HIV mice it was reduced, but the combination and individual treatments appear to rescue the impairment in eco-HIV mice. And finally, we did here catwalk gait analysis. Again, in the case of catwalk gait analysis, there are like over 50 parameters. And we don't know what is important, what is not important. So what we looked at all these parameters and then came up with some of the parameters which were really relevant for our study, for hand-related study. So we, at least in this study, we looked at the contact area, we looked at average speed for running, and the print area. And what we found that in all these cases, in HIV mice, they were reduced. And then in the case of treatment, they were pretty much either significantly or appeared to have rescued these effects. So clearly, even catwalk analysis suggests that there is some reduction in certain parameters, and that can be rescued by the combination treatment. We looked at inflammatory response as well, and same thing in the HIV mice. Many of these inflammatory cytokines are enhanced, like IL-6, IL-1 beta, RANTage, and with the treatment, it was reduced to close to wild type or similar to wild type level in all these cases. We also did oxidative DNA damage, where we saw that DNA damage in the brain cells was induced in the case of HIV mice, and the treatment has significantly reduced, and pretty much to the level of non-infected mice. So all these studies again showed that clearly this combination can reduce inflammation as well as oxidative stress, which are induced by HIV. Similar findings we also saw in the systemic plasma, but the magnitude of the changes was less than the brain cells. So overall, what we concluded from this study that curcumin treatment can enhance the EVG concentration not just in macrophages but also in mice brain. It can reduce inflammation and does not change neuronal homeostasis. In the case of behavior study, we also found out that HIV can alter behavior study as per determined by NOR or Morris Water maze or catwalk analysis. And then these are further rescued or brought back to the non-HIV mice level when we treat with curcumin and darunavir, in this case, darunavir together. I believe it's the EVG, not darunavir. In the case of inflammation, same thing. It was increased with HIV, and then together it was reduced. So clearly, we have shown so far that the optimal route for drug administration could be intranasal, but at the same time, nutraceutical can have also impact in terms of adjuvant therapy, because we saw reduction in behavior impairment, reduction in inflammation, oxidative stress when we have this combination therapy. So now our next goal, and the main goal, is to use of nanoparticles. Several years ago, we started this study with synthetic nanoparticles called PLGA, and most of us know that PLGA is a FDA-approved nanoparticle, which has been used for many treatment strategy, mainly for cancer and peripheral diseases, but not for neuronal diseases. So we were trying to use this nanoparticle for hand as well. So we were able to encapsulate at least two different ART drugs, elbitagravir and darunavir. That size in terms of TEM is shown at least for one of them. And over the period of last several years, we have published many papers on PLGA EVG nanoformulation and PLGA DRV nanoformulation. And in general, what we have shown that these nanoformulation can enhance delivery of these drugs in the in vitro macrophages as well as blood-brain barrier model and also in HIV mice. And they not only just increase the concentration, they can reduce neuro-HIV as well as HIV-associated inflammation. So that's kind of summary of all these studies. Now, very recently, we just published this paper last month, where we looked at now PLGA nanoparticle encapsulated with both elbitagravir and curcumin. And we wanted to see when we have a combination therapy with elbitagravir and curcumin in PLGA encapsulated system, will we have better effect? So you can see that we encapsulated curcumin here. We have very similar encapsulation efficiency, about 90%, which we already published before by others as well. Importantly, that when we have this EVG and EVG curcumin combination therapy encapsulated in PLGA, we again have increase in EVG concentration. And when we look at the area under the curve, this was also increased significantly. We looked at the same in a wild type mice using IP study in this case. And what we see that clearly the combination therapy with PLGA, the plasma concentration of EVG is increased, not just with early time point, but overall area under the curve as well. It was also increased in brain, lungs, and liver. So clearly PLGA is facilitating delivery of EVG, and the presence of curcumin, which we have shown before also, it further enhanced the concentration for EVG drug. We looked at some of the neuroinflammatory proteins, and we could see that in both E1 cells and wild-type mice, there's a reduction in several HIV-induced inflammatory markers like TNF-alpha, IL-1 beta, IL-18, with either individual drugs or with combination. So clearly in both E1 cells as well as wild-type mice. So clearly EVG and curcumin combination worked, which we have shown before, but when we package those in synthetic nanoparticle, PLGA, we can further enhance drug delivery and reduce inflammation. So far, we have shown that PLGA-based drug delivery of EVG, DRV, and combination can enhance drug bioavailability and suppress HIV neuropathogenesis. But of course, there are certain limitations with that. And the limitations are that intranasal delivery is an issue with PLGA, where PLGA does not form a good solution. It's more like a suspension. So the intranasal drug delivery becomes challenging. Second is a biodegradability and disposition from the brain. But even though PLGA is FDA approved, they have not been used as a drug for neuronal diseases. And the drug targeting could be another issue unless we engineer PLGA with a certain drug targeting moiety like antibody or other proteins. So what next? So we thought, let's think about natural nanoparticles, based drug delivery, which is exosomes or extracellular vesicles. As most of us know now, that exosomes are produced by pretty much all kinds of tissues and organs, and they are secreted in plasma or CSF, and they circulate throughout the body. So they can carry certain biomolecules from just a peripheral system to brain and vice versa. And that could be for good reason or bad reason, depending upon the trigger. So in last many years, what we did, we studied the cell-cell interactions via exosomes, which are in the presence of different drugs of abuse, like alcohol or tobacco. We also studied whether we can find certain EV-based biomarkers, which are altered in the presence of upon tobacco-induced HIV pathogenesis or alcohol-induced HIV pathogenesis. So we did find certain biomolecules, either membrane, either neuronal proteins or even plasma proteins, which are altered in different conditions. So clearly, EVs can be used to study cell-cell interactions as well as a bio signature or biomarker. But that's not the what I'm going to talk today. That's a kind of talk for some different time. Today, I'm going to focus on EVs for drug delivery. So what are the benefits of EVs for drug delivery? The benefits are clearly inherent biocompatibility because these comes from natural sources. It also has appropriate size and distribution in the body, within 200 nanometer, and it can also have capability for intrinsic targeting, specifically macroglial homing. In fact, the general idea is that if you generate EV from certain source, certain cells or cell line, it is going to go and target back to the same cells or cell line or similar cells. But there are certain limitations as well. For example, we don't know what are the EV's components and how do they interact with our drug if we encapsulate our drug. We do not know the short, they have usually short half-life, and their half-life can vary based on the sources of the EVs. We also do not know what kind of unexpected immune activation or inflammatory response may occur. We have no idea about this. So these are the limitations that have to be figured out as we move forward, for human or clinical trial. So very simple strategy. I worked with Dr. Kochat at our PLAS Center for Drug Delivery Solution, and also our postdoc Asit and students Sandeep and Lina. And the strategy is very simple. We isolate EVs from certain cell lines which secrete in the media. We load drug into those EVs, target if necessary, do biodistribution study, mostly in wild-type mice, and then finally, pharmacodynamic study in eco-HIV mice or other humanized mice, and then clinical trials. So that's a simple strategy which we want to utilize. So the first thing is isolation of EVs for optimal drug loading. So we used multiple methods like precipitation, gel filtration, and ultracentrifugation. But we found that the ultracentrifugation method gave the best outcomes. Even though ultracentrifugation methods are not the best when it comes to yield is very, very low, but at least the quality is really good in terms of drug loading. Then we optimize drug encapsulation efficiency using multiple methods. First, we use endogenous method, where basically you treat cells, certain cell line with drug, and then after a few days, 3 to 4 days, you isolate EVs from the media, and that may have drug loaded inside the EVs. Then we also did exogenous method, where we isolated EVs and then use different method like probe sonication method or water bath sonication method to load our drug. And upon all these methods, we found out that probe sonication methods are the best method to load most optimal drug. So but the problem was our loading efficiency was very low. When we have now on, I am going to use the code word for the source of EVs, drug, and also nutraceuticals. The reason is that some of these have some ability to be patented, and we have done that, provisionally patented at least one of the study. So we are going to just use the code word. As I said, the problem is the loading is very, very low. It's less than 10%. And we know that with the synthetic nanoparticles, we had about 90% loading. So clearly there's an issue, and 10% loading may not be sufficient. We did some BBB permeability study with even low loading, and we could see that when we have ART01 with the extracellular vesicles, we are able to enhance the BBB permeability. And not just in the media, but also in the macrophages, which are other side of BBB in vitro model. So clearly, extracellular vesicles loaded drug can have better delivery capability across the BBB. But the problem again is that the loading is not sufficient to do subsequent animal experiments, especially via intranasal route. So we have to find another way to either enhance the loading or maybe find some kind of hybrid system. So hybrid system is basically fusing artificial nanoparticles and natural nanoparticles together. And both have their own advantages. For example, in the case of artificial nanoparticles, if you can have improved drug loading and slow drug release. While in the case of natural nanoparticle, we can have cell targeting and they are more biocompatible. So we can mix these two, we can improve both PK and PD. That's the overall goal. Because none of them are alone really the best, best for our purpose. So the way you do fusion is this, that you prepare lipid nanoparticle, you load drug inside, then you prepare EVs, and then fuse these two together using different method, which can distribute the drug throughout the EV lipid nanoparticles or EV liposomes nanoparticles. Making long story short, we optimized EV preparation for our hybrid system, and we found out that gel filtration and precipitation method were best for this purpose. And since we are not looking for intact EV in this purpose, gel ultracentrifugation were not really needed in this case because we are breaking these EVs anyway. Then we found out the source of EVs, EV01 was the best source of EVs. Out of 3 different sources we looked at. ART drugs, we studied with ART01 and ART02, and nutraceuticals 01. We used different membrane fusion method for drug loading. We found extrusion was the best method for optimal drug loading. And then we characterized in terms of physical characteristics and also in encapsulation efficiency, stability, drug release, etc. So here's the first most exciting result. We found encapsulation efficiency up to 40%. If you remember, our EV alone gives less than 10%, and lipids like PLGA give almost 90%. This is somewhere in the middle when we have the hybrid system. That makes sense, kind of. We characterize this hybrid system in terms of TEM and then zeta potential and zeta size. And clearly, there's not much change in terms of size or zeta potential when we have ART02 loaded hybrid system. Our physical stability was also really good, especially when we have loaded drug. When the drug is loaded in EVs, the stability is even better compared to EVs alone at both room temperature as well as 4 degrees. We looked at the typical EV biomarkers, CD63, ALEX, and CD9. They were all present. We looked at drug release. Unfortunately, the drug release was quite fast. Half-life was 3 hours or even less, and almost 95% was released within 24 hours. They were hemocompatible. Even at 60 microgram per ml drug, which is really very high, it only shows 5% or less hemolysis. So clearly, this is stable in our blood. Then we looked at biodistribution in wild-type mice. Very similar experiment what we did before with the other drugs and combination. And what we see in this case, that in the case of especially intranasal drug delivery and with the EV-loaded drug, clearly there's an increase in brain-to-plasma ratio. Although only brain did not increase much, but in the case of brain-to-plasma ratio, which may be a better characteristics of having a drug in the brain and at the same time having less off-target effect. So clearly it's higher. Even though with IN at half the concentration, we have similar or better permeability in the brain and brain-to-plasma ratio. And they were reduced in terms of in lungs and liver. So clearly the off-target effect is less when we have EV-loaded ART02 drug and in the presence of intranasal drug delivery. There, as previously described, we studied behavior, hand-associated behavior, and in this case, we studied NOR and cat-walk gait analysis. So in terms of NOR analysis, clearly, as we know that in the case of HIV, both discrimination and recognition index are reduced. But when we have HIV treated with this drug, ART02 drug, loaded in EV lipid hybrid nanoparticle, both IP and IN administration. So clearly, the loaded drugs and in the presence of IN administration, they rescue recognition index and to some extent discrimination index as well. We did cat-walk analysis, very similar. Here we looked at these 4 parameters, which is swing speed, print width, coupling in both sides, left hind and also to front and right. What we see that in all these cases, HIV reduces like swing speed or pin width. But when we have the treatment with EV lipid hybrid system loaded with ART02 and with intranasal drug delivery, clearly there's a rescue of HIV-associated swing speed or pin width. Even coupling was also rescued. In one case, left side of the body, the coupling was reduced, but the right side of the body, the coupling was increased, but both were rescued back to pretty much non-HIV level. So that was again highly significant data we could see from this study. We also looked at the inflammatory markers, mainly IL-1 beta and IL-6. And what we could see that IL-1 beta and IL-6, although both are increased with HIV, but with treatment, they are rescued or brought back to the non-HIV level. DNA damage, very similar result. That with the treatment, we have much lower oxidative DNA damage, specifically when we have hybrid system and intranasal drug delivery. So overall, from this study, we could also show that the brain concentration, both IP and IN, are increased when we have hybrid system with ART02, and lungs and liver concentration are decreased. Means the off-target effect is less. So that's very, very important to know that we need our drug to reach brain as much as possible. At the same time, they do not accumulate in lungs, liver, and kidney, that may cause toxicity. Our behavior study and other neuropathogenesis study have also shown that HIV have reduced like both NOR, recognition and discrimination index, and in catwalk study as well. But the treatment with the hybrid system loaded with ART02, specifically intranasal, have rescued it. Same thing happens with inflammatory response and oxidative DNA damage. So clearly this formulation, hybrid formulation with at least one of the drug and intranasal drug delivery are beneficial. Now, in last few months, which is ongoing study, we started working with different drugs and different nutraceuticals, as well as combination therapy. So what you see here, that another drug called ART01 and nutraceuticals NC01 and a combo therapy. So in this case, we did not use these two drugs we did not formulate these two drugs individually. We wanted to formulate together. So ultimately, if this goes to human, they may have only one pill or only one delivery, not like two different drug formulation. So we made combo. The size of all these are very similar, not much changed. Interestingly, the encapsulation efficiency has gone up significantly. If you remember, or if you hear also for individual drug ART, it only showed about 40% encapsulation efficiency. Now we see almost 60%, which is 50% increase in encapsulation efficiency in these drugs when we have combination. And stability is also increased. In the previous single drug, we saw up to 40 days for stability at room temperature and 4 degrees. In this case, up to 70 days at both room temperature and 4 degrees Celsius. The combination is very stable. At the same time, the release profile is become now biphasic in this case than compared to the other one, which are more like a Meckless-Menten kinetics. It's a biphasic. And importantly, the half-life has increased from 3 hours to 10 plus hours, up to almost 20 hours for nutraceuticals when we have combination therapy. So this is very, very exciting early result where we are able to make combination therapy in hybrid system with at least one drug and one nutraceuticals. And that combination therapy can enhance encapsulation efficiency, stability, and drug release profile. Now we are in the process of studying other biochemical and biophysical properties. We are going to study, of course, biodistribution in wild type, and then HIV neuropathogenesis and hand in eco-HIV mice. So I hope overall we have convinced you that we are able to produce EV lipid hybrid system, not just for ARTs and nutraceuticals, but also for combination formulation. These hybrid systems, at least in the single drug, can increase brain drug concentration, can improve cognitive and motor function, and reduce inflammatory response and oxidative stress. However, this is not the end. We will continue to improve the encapsulation efficiency. A goal is if you can reach to 80%, that would be really good by maybe changing the composition of different lipids and changing maybe the source of EVs and also composition of EVs. That may also improve drug release profile. The goal is to have slow release profile, not just 20 hours, but maybe 2 days or 4 days. And then we need to improve the translation from mice to human as well. Because if you look at the intranasal drug delivery, both of these animals have different kind of nasal surface. Okay? So we have to work on that as well. So our future study ultimately will be again to have comprehensive PK and safety study and safety profile. We have not done safety profile yet. We need to make sure that for 3 to 6 months, when we treat with these drugs, at least in wild type to begin with, there is no safety issue. We will study the mechanism of EV lipid nanoparticle mediated effect. We will also ensure the large scale preparation using some kind of bioreactor system, which we are using also at this point of time. And ultimately, our formulation has to be translated from animal to human drug delivery. So we are going to work on that in future. Again, it all depends on how the funding going to be, because if there's no funding, of course, we can't do any of those studies. And as we know that it's not the great situation at this point of time for HIV research. But anyway, so I hope that I have been able to show evidence for our strategy, which utilizes 3 different ideas. The one is the administration route, intranasal. Second is nutraceuticals, different kind of drugs we have used. And then third is nanoparticle-based drug delivery. In our case, finally, we came up with hybrid drug delivery. And all these have their advantages compared to just single drug, which we use today in conventional way, either oral or IV. So I hope that this will continue in the future, and we'll have more data maybe to show by next year. Of course, these data or these studies would not have been possible without the support of a lot of students. In fact, since the beginning of our lab, we have had at least 11 students, grad students, who contributed to different work in our lab. Several postdoc fellow as well. Some of the student and postdoc fellow I have highlighted are the one who did this study, and that I presented just now. And many collaborators over the years, and mainly the one who are who are highlighted. They are the one who contributed to this current work. So thank you, everyone, all the collaborators, students, postdoc, and of course, NIH funding. And I hope that we continue to get funding so we continue to advance science in terms of hand treatment in the future. So thank you very much for being a nice audience. Clearly, I think we have almost 15 minutes for any questions. Yeah, I think Jules Levine has a question.
Yes, hi. Thank you for your research and your presentation. Several studies have been reported showing that dolutegravir levels increase in older people with HIV, and when levels of dolutegravir are higher, it can cause psychiatric side effects. So I know you looked at elvitegravir, but with dolutegravir, there's this concern that you increase the levels, which you would, I guess, with curcumin, and you risk the potential for increased psychiatric side effects. So what are your thoughts about that?
Thank you. That's an excellent question. And yes, I have seen some report as well as seminar where dolutegravir clearly have some side effect in terms of psychological or psychiatric, and even raltegravir, which are similar class, may have similar. In fact, that's one of the reasons why we have not started with those drugs yet, because we know that those drugs are not well studied yet when it comes to neuronal impact. So I don't know what to say, but goal is if we use those drugs for our purpose, we have to ensure that we reduce the drug dose, maybe with the help of adjuvant therapy or with the help of other which are low drug but still effective. But again, at this time, I really have no answer for that question. But that's a great concern we have with those new drugs. Thank you.
I think Smita has a question.
Sure.
Thank you, Matthew. Hi, Santosh. Really nice talk. Trying to turn on my camera. I had a question about one of the initial data you showed comparing IN versus IV. I'm assuming this was not aerosolized delivery.
Which delivery you said?
Was this aerosolized by any chance?
No, these were we studied IV and IN delivery.
Okay. And then have you looked at regions of the brain more distal from the olfactory bulb to compare them in IN versus IV? And my second question is, have you looked at the CSF? And then a final question, it seemed like you had an initial rise at the early time point with better levels with the IN, but the trough concentrations seem to be similar. So if you can comment on that, that'd be great. Thank you.
Great. So clearly, those are some of the things which we are planning to do in our future study. We have not looked at different region of brain yet and CSF. But that's a very good question because clearly we have to ensure that those reaches the target where HIV infected cells are there. And the last question you talk about, remind me again.
I myself have forgotten it, but this leads me to another question, which was, at least with infectious virus, we see better tropism or better viral infection with aerosolized versus just application in the nares. So that's something you might want to consider. Oh yeah, the trough levels. My third question was the trough levels. Yeah.
Yeah. So we have a similar trough level, and again, I mean, at this point of time, we can only say that yes, immediately it goes to brain, lot more than this, and then it taper down in both the cases. But this is just again couple of drug doses we have used right now that need to be studied at multiple drug ultimately, and also with our formulation, because our ultimate goal is with formulation, because that's where we want to have slow drug release. And it's possible that when we have slow drug release with formulation, our immediate drug concentration and then trough may not be the issue. It may be, it may be very consistent in the brain for longer period of time, which are effective and not neurotoxic. So that's kind of the ultimate goal with our formulation.
And when you say brain, which region of the brain are you looking at?
So we are at this time, we are looking at whole brain. We are not looking at any specific region yet.
Okay. All right. Thanks for clarifying.
Yeah, sure, Smith.
We have a few questions from the chat. One of the questions is from Barbara Slusher. Is the loading efficiency dependent on the physiochemical properties of the drug being loaded?
That is true. So it is a characteristics of, I would say, 3. It's a composition of, so what kind of lipids we are using. So we are not right now talking about telling you the lipid we are using. What is the ratio between lipid and exosomes? What is the source of exosome? And of course, drug itself. The good thing is the majority of the drug I have not shown that, but when we compare the drug characteristics which we are trying to use, mostly protease inhibitors, they are very and integrase inhibitors, they are very similar characteristics when it comes to PK and the half-life, stability. So I believe that between these drugs which we are trying to use, which are integrase inhibitors and protease inhibitors, that impact may not be as great as when we are talking about composition of different lipids and different, exosome, etc. But yes, that is the consideration when you have drugs of two different types altogether.
Just this Barb Slusher, I asked that question. Can I just follow up? Do you have a sense of why the efficiency is so different between the EVs and the nanoparticles? Do you understand why you're getting such a big difference there, and why when you combine them you get something intermediate?
I mean, the one simple thing I can tell you that maybe in the case of synthetic, you have a lot of rooms inside the vesicles to encapsulate drugs. Even though those drugs may be also on the membrane quite a bit because they are lipophilic drugs, but they may also be inside. But in the case of EVs, maybe they are not able to go inside the EVs because they have a lot of other components which are there in the present. So that could be one of the reason. It's just the physicochemical characteristics of these two. But when we combine, we are kind of compromising on both. In one case, we are having, because when we, so when we load with lesser lipid, we load with 90%, right? And then we fuse with exosomes.
Right.
Then when we fuse, it's like some of the drugs are eliminated or removed as a result of these EVs. So again, that's one of the things which we have to do, the interaction between the component, lipid and EV components and drugs. How do they interact? That's something which is really going to be exciting, biochemical characterizations of these nanoparticles.
Thank you. Thanks.
Yeah, thank you.
Okay, another question from the chat from Ron Reyes is, how do you confirm that the drug is not adhering to the surface of the extracellular vesicles and is encapsulated efficiently?
Again, at this time, we have only done FTIR study just to show that here they fuse and the drugs are there because we measure drug concentration after that. We don't know how much drug is just on the surface, how much inside the membrane, and how much is inside the vesicles. We don't know at this point of time. Those are definitely detailed characterization we have to do once we have, I would say, the most optimal system.
Satosh, can I ask a follow-up question on this? Once you fuse the EVs with the PEG nanoparticles, you're getting better efficiency, but your PK did not really change. How do you know that this fusion of the drug delivery systems will be targeted to the brain better than individuals? Like, what is the purpose of fusing the two if you're already getting good loading efficiency? And what is the mechanism of drug release eventually once it gets to the brain? Because you're measuring the whole homogenate in the brain where everything will be broken down. So you don't know if it's still encapsulated or is it the free drug? How do you try to differentiate that?
Yeah, so all those are really great questions, and most of these study will be done in terms of targeting. Based on the characteristic, we believe that when we have the hybrid system, our targeting will be better because we know that EVs coming from a particular source can target similar cells in the brain. So I can give you, we have tried, let's say, plasma-derived EVs. We have tried macrophage-derived EVs. We have tried HMSCs, stem cells-derived EVs. And some of these are known to target certain brain cells. Others have shown, we have shown. But we have not studied that yet inside the brain, that where exactly they are targeting. And in terms of drug release, the drug release may be similar. So our goal is that we achieve the drug release as good as synthetic nanoparticle. Because synthetic nanoparticle drug release are really good. It can go up to several days. But with EVs, we saw very fast. But with hybrid system, our goal is to have somewhere intermediate release where we can go over 1 day at least half-life. We have already achieved up to 18 hours half-life, at least in one case, but goal is to have more than a day half-life. It won't be as good as PLGA, we know, but the targeting will be better. That's kind of our expectation. And all these we have to finally show in this study.
Thank you.
Thank you.
There's one more question in the chat from Mei Huang. Which brain regions did you measure cytokine levels in? Was that also whole brain?
Yes, it's also at this point whole brain, cytokine, chemokine, either using ELISA or Western blot. Yes, we have not looked at individual site yet.
I have a follow-up question. So, did you see any inflammation while you're applying EVs to the mice?
So inflammation in terms of just with EVs, right?
Yeah.
If I remember some of those data, just with EVs or even hybrid system, we did not see much inflammation. In fact, in some of the study what we have shown, even in PLGA, that actually inflammatory cytokines and chemokines levels were reduced with just either PLGA or EV system. Now, that could be a good news, but we don't know how it does it. But actually, we saw some reduction rather than increase in inflammation.
Okay, yeah, thank you.
Yeah. I think, Barbara, you have a question also?
Yeah, just actually one more question. Have you, we've done a fair amount of intranasal studies, which are kind of hard in the mouse with putting the pipette in the nair. Have you tried any of the devices? Like I know we've worked with Impel, which will actually move the drug to the latter part of the nose to try to bypass the respiratory epithelium so you get more brain delivery. Have you attempted to use any of those types of devices to improve your brain to plasma ratio?
Not yet. At this point of time, we are just making this solution and kind of, yes, with pipette, like a drop by drop up to 25 microliter at one time. That's what we are doing.
Yeah.
But you are right. The goal would be that to formulate these in a way that we can use some kind of device using aerosols or some kind of those system. Because that will be also more useful and easy to handle ultimately.
Yeah. Okay.
So thanks for, probably we'll get back to you and see what kind of devices, we have tried and others have tried, so we can move into that as well.
Yeah, for sure. Thank you. Really enjoyed your talk. We do a lot of drug discovery and drug delivery and intranasal administration. So it was great to hear your talk today. Really appreciate it.
Thank you very much. I appreciate it.
Thank you very much. Is there any other questions? Okay, well, thank you, Santosh. Thank you for that wonderful talk. And it's very nice to have you today.
Thank you. And everyone, this is right 12 o'clock my time, 1 o'clock your time. So perfect. Okay, have a great day.
Carolyn, do you
Okay.
want to tell us what's up next week?
Yeah, so we don't have a seminar next week on Wednesday because we have the symposium, JHCon symposium. It's Modern Translational Approaches to NeuroHIV, and it's on Tuesday the 8th from 9 a.m. to 3 p.m. Eastern. And if you're at Johns Hopkins or nearby, you can join in person. We're having it live or virtually via Zoom. And I'll send another email letting everybody know how to RSVP.
Yeah, thanks, Carolyn.
All right.
Thank you.
All right. Thanks, everyone.
Thank you.
Bye.
Bye.